All right, we have most of our registrants online over here today, so we will start our webinar. So good afternoon to everyone once again, and welcome to our webinar with the title Bispecific Antibody Overview and Production with Crossmap Technology. We are thrilled to have you to join us today as we introduce the advantages of bispecific antibodies, their mechanism of action, and the development history of bispecific antibody platform. So before we start our webinar, I would like to introduce our speaker of today, which is Dr. Wang Sai, our senior scientist from GenScript. So Dr. Wang Sai was trained in the field of structural and molecular biology. She joined GenScript Singapore Protein Production Department 3 years ago as a protein senior scientist. During her career in GenScript, she provides professional evaluation and advises for various projects on a daily basis. Dr. Wang Sai is also one of the key players in GenScript bispecific antibody R&D project and establishment of bispecific antibody production platform. We will cover the following topics in today's webinar, which is an understanding of bispecific antibody, an overview of bispecific antibody mechanism of action and development history. We will also share some case study on Crossmap production. If you have any questions for our speaker, please leave your questions at the Q&A box and we will answer them after the presentation. This webinar will also be recorded and you will have access to the recording after the webinar as well. So without further ado, let's welcome our speaker, Dr. Wang Sai. Dr. Wang, the floor is yours.
Hello everyone. Thank you for the kind introduction, and welcome to the webinar. And the topic today is bispecific antibody overview and production with Crossmap technology. Here is a preview of today's talk. I'll first give you a brief introduction of bispecific antibody, which will then be followed by examples of different mechanisms of action, followed by the development history of bispecific antibody platforms, and a list of commercialized bispecific antibodies. And in the later session, I'll be focusing on Crossmap technology and showcasing production of 2 Crossmap bispecific antibodies by GenScript mammalian recombinant protein production platform. And last but not the least, I'll give you a quick introduction of GenScript TurboCHO mammalian platform services. So first, a comparison of monoclonal antibody and a bispecific antibody in some of the features. So a typical monoclonal antibody targets one epitope. The OKT3 is the first monoclonal antibody approved by FDA in 1986. And it targets the CD3 on T cells and effectively depletes the T cells from the circulation. So in making it useful in preventing and treating acute rejection in organ transplantation. A bispecific antibody is basically an artificial protein. It has 2 distinct binding domains that can bind to 2 antigens or 2 epitopes of the same antigen. Over the past 2 decades, bispecific antibody development has been revolutionized due to genetic engineering approaches that enable a wide variety of molecular structures that offer different advantages. By targeting 2 antigens or epitopes, they can cause multiple physiological responses, which may be independent or connected. These treatments may act like a cocktail of 2 monoclonal antibodies, but drug developers need to manufacture only 1 molecule, and the patients may need only 1 antibody treatment. Plus, their synergistic features may produce more significant treatment effects. Currently, most bispecific antibodies in development aim to treat cancer, but others are focused on chronic inflammatory, autoimmune, and neurodegenerative diseases, and vascular, ocular, and blood-related disorders, and infections. So now let's take a look at some of the mechanisms of action of bispecific antibody. The majority of bispecific antibodies in clinical trials are CD3 T-cell engagers. And these molecules bind to CD3 expressed on the surface of T cells and to a tumor-associated antigen on the tumor cell surface. When both CD3 and the tumor-associated antigen are engaged, the proximity of the T cell and the tumor cell result in the formation of an immune synapse and the stimulation of the T cell, redirection of the cytotoxicity against the tumor cells. Another mechanism is called through bridging of two different receptors on one cell. So in which the cross-linking of specific cell surface receptors leads to their inactivation or activation. So in the case of inactivation, one example is to reduce the tumor growth in cancer applications. And in the case of activation, one example is that the activation of a growth factor receptor for the treatment of diabetes. And bispecific antibody may also be designed for the precise positioning of an enzyme and a substrate as a cofactor mimetic. So for example, the bispecific antibody emicizumab is used to replace a critical clotting factor, the factor 8, in the treatment of type A hemophilia. So in all this discussed the mechanisms, binding of the bispecific moieties occurs simultaneously. And there are also mechanisms of action in which binding of the bispecific moieties occurs sequentially. Piggybacking allows the translocation of one active binding arm to an otherwise inaccessible compartment, such as central nervous disease targets protected by the blood-brain barrier, or bacteria and viral antigens with a role in endosomal escape. To date, more than 20 different commercialized technology platforms are available for bispecific antibody creation and development. The original concept of bispecific antibodies was proposed by Lisanov and his collaborators in the 1960s, including the first idea of antibody architecture and the first fragment-based format. In 1975, the problem of producing pure antibodies was solved by the creation of hybridoma technology, and the new era of monoclonal antibodies came. In 1983, hybrid hybridoma or chondroma technology was created. And in 1988, the single-chain variable fragment, SCFW, was invented to minimize the chain association issue. And in 1995, the first solution to light chain association issue was developed through species-restricted light chain pairing. In 1996, the bispecific antibodies became more developed when the knock-into-hole technology emerged. Heavy chain association issue was minimized through the use of complementary heavy chains. In 1998, common light chain was first used to tackle the light chain pairing problem. And in 1999, it was discovered that natural human IgG4 is bispecific. And the following two highlights were in 2007, which are the discovery and elucidation of bispecific human IgG4 Fab-arm exchange process in vivo, and the invention of dual variable domain IG symmetric format. And in 2011, CrossMap technology using the domain crossover was developed to solve the light chain association issue. And I will further elaborate on Crossmap in the second part of the talk. And in 2013, bispecific IgG1 was generated through Fab-arm exchange of separately expressed antibodies. And in 2014, orthogonal Fab interface was first discussed as a solution to light chain association issue. Currently, there are 13 bispecific antibodies approved worldwide, and among them, 11 were approved by FDA, as highlighted in blue. And as you can notice, majority of them, in total of 10 bispecific antibodies, were approved in the last 2 years. In the next session, I will be focusing on CrossMab bispecific antibody technology, which were applied in the development of glofitamab and bsAb. As has been briefly introduced earlier, CrossMap technology was first described by Roche in 2011 as a novel approach enabling cracked antibody light chain association with their respective hepatitis chain in the bispecific antibody. Since the original description, CrossMap technology was evolved in the past decade into one of the most mature and broadly applied technologies in the field. And dozens of bispecific antibodies based on CrossMap technology developed by Roche and others have entered the clinical trials. The most advanced of these are the glofitamab and bsAb. Both have been approved by FDA and EMA in the past 2 years. So the co-expression of 2 different heavy chains and 2 different light chains result in a complex mixture of 16 possible recombinations, representing 10 different antibodies shown here. And only one of these antibodies corresponds to the desired bispecific antibody. And the maximal yield of which is only 12.5% in the mixture. So 2 problems must be solved to produce the desired bispecific antibody exclusively. So first, the effective induction of the heterodimerization of the 2 heavy chains and discrimination between the 2 light chain heavy chain interactions. Correct heavy chain association resulting in a heterodimeric Fc can be enforced using the knob into hole technology by introducing a bulky tryptophan residue in one Fc fragment and forming a corresponding cavity on the other Fc fragment that can accommodate this tryptophan residue. And more recently, multiple alternative approaches to enable correct heavy chain association have been described, such as relying on the charge interactions through the electrostatic steering strategies. Although knob into hole technology was developed in the late 1990s, enabling correct light chain association remained a major problem. And the only approach to achieve this before CrossMab was invented relied on the use of common light chains for both specificities. However, the use of a common light chain requires the de novo identification of the corresponding antibody pairs, which can be challenging and time-consuming depending on the desired type. And it restricts the availability and diversity of antibodies that can be used. So that methods allowing the generation of bispecific antibodies from pre-existing antibody pairs were highly desired. So CrossMap technology innovated a domain crossover strategy to enable correct light chain association in bispecific antibodies. Here goes the basic principle. So the original heavy chain VH-CH1 domains were incorporated as the novel light chain, and the original light chain VL-CL domains were fused to the hinge region of the Fc fragment as the novel heavy chain. So correct heavy chain association can be enforced in this Fab crossover format. And this strategy has also been described as a Fab in tandem IJ format. So in addition to Fab crossover, there are two other formats. In Crossmap VH-VL format, only the VH-VL domains are exchanged. Alternatively, in the CH1-CL format, only the CH1 and CL domains are exchanged. And in the case of CH1-CL crossover, no theoretical side products due to the domain crossover is expected, and the crystal structure analysis confirmed the structural integrity of the cross the Fab domain in this format. And in the case of Fab crossover, two heavy chain and light chain based side products can be observed. However, the correct preferential formation of the Crossmap Fab can be fostered by relatively overexpression of the respective light chains, so that respective undesired monovalent and binding inactive side products do not form in significant amounts. And similarly, in the case of VH-VL crossover, a Bence-Jones-like side product based on VL-VL together with the CH1-CL interaction can be observed. And in order to avoid the formation of the side product, natural charge pairs in the Fab were identified and the respective orthogonal charge interactions were introduced into these non-exchanged antibody CH1-CL domains. As a consequence, the undesired Bence-Jones-like side product does not form due to the repulsive charge interactions, whereas the desired light chain pairs, they can pair correctly in the non-crossed Fab due to attractive charge interactions. And in the following slides, I will be showcasing production of two Crossmap bispecific antibodies by GenScript mammalian recombinant protein production platform. So first is glofitamab, which was sold under the brand name Columvi. It is a bispecific CD20 directed CD3 T-cell engager developed by Roche. Glofitamab received the approval in 2023 for the treatment of adult patients with a relapsed or refractory diffuse large B-cell lymphoma, or it's called DLBCL. And let's take a look at the details of the engineering of the molecule. Glofitamab, first it has a 2-to-1 configuration with a bivalency showing here in red and orange targeting towards CD20 and the monovalency towards CD3. And it works by recruiting B cells directly to the surface of cancerous B cells. And the correct heavy chain association was enforced by knob-into-hole technology, and the correct light chain association was enabled by CrossMap VH-VL domain crossover. And in order to avoid the formation of Bence-Jones-like side product, the orthogonal charge interactions were introduced into the non-exchanged antibody CH1-CL domains. So the antibody sequences were taken directly from the publication, and gene coding sequences were optimized by GenScript proprietary codon optimization algorithm. And after an initial small-scale chain ratio screening, 1 liter scale-up production was done using the turbo-control transit expression system. The clarified cell culture supernatant went through 2 steps of chromatographic purification using protein A affinity method, followed by size exclusion method. And quality controls was done by diglycosylated mass, SCCHPLC, and CDSDS. The yield of purified antibody was 24 milligrams per liter of cell culture, and purity was over 95% indicated by CDSDS and over 99% indicated by SCCHPLC. And endotoxin level was below 0.1 EU per mg of purified antibody. And in this second case study, the antibody molecule takes the 1+1 CrossMap CH1 and CL crossover configuration. So the correct heavy chain association was again enforced by Nobel two-hole technology, and the correct light chain association was enabled by Crossmap CH1 CL domain crossover. Again, antibody sequences were optimized. The gene sequences were optimized by GenScript proprietary dual-tree codon optimization. Expression was done in 100 ml turbo-control transit system. And the cell culture supernatant went through 2 steps purification using protein A affinity chromatography, followed by capture/kappa affinity chromatography. And after the first step purification by protein A, the SDS page and the HPLC suggested over 96% purity. However, the high resolution reverse phase liquid chromatography suggested the presence of light chain mispairing, and this impurity accounted for around 18% of the total product. So capture/kappa was chosen for the second step of purification based on the differential affinity to the purification resin between the protein of interest and the light chain paired impurity. And eventually, the purity was increased to about 95% by both SDS page and SCCHPLC. And the light chain mispairing component was completely removed. And the yield of purified antibody was 189 milligrams per liter of cell culture. So we are almost to the end of today's talk, and a quick summary of what have been shared. First, bispecific antibody and their mechanism of action were introduced, followed by a chronological review of the development of bispecific antibody technology platforms and the overview of commercialized bispecific antibodies. And we then move on to the introduction of Crossmap technology in more details, followed by 2 case studies of Crossmap production by GenScript mammalian platform. And GenScript is providing proprietary turbo-control platform, which offers high yield performance within a short turnaround time. And this cost-effective production system enables small to large-scale production of antibodies with a high level of consistency, making it an excellent complement for target discovery and pre-evaluation pipelines. And GenScript can handle your bispecific antibody project from micrograms level to grams level seamlessly. And empowered by the strong R&D capability, turbo-control platform provides unparalleled services for bispecific antibody production with specialized format design, AI-driven codon optimization, optimize the plasmid ratio and the production process, and specialize the multi-step purification methods. And Bispecific is providing an updated TurboCHO bispecific antibody web page. If you scan the QR code here, you will see the details, and the highlight is the interactive bispecific antibody table. And if you click on a pictogram, you will see more details of the molecule, namely the manufacturability of the format, recommended purification methods, and its typical yield. So this comes to the end of my talk, and thank you for watching. And I'm happy to discuss any questions related to this webinar.
All right, thank you, Dr. Wang. We do have some questions from the participants, so we'll go through them one by one right now, okay? So the first question from Tilina, I'm rephrasing over here, but I think she is asking, is bispecific technology similar in base on hybridoma technologies?
So from my understanding, hybridoma technology is basically used for the production of monoclonal antibody. And yeah, so the Crossmap and our bispecific antibody platforms are developed for the production of the given sequences of the bispecific antibody molecules. So they are basically in different stage of the antibody discovery technologies.
All right, thank you, Dr. Wang. So the next question, what is the chain ratio screening and how do you do it?
So basically, the reason we do the chain ratio screening at the first stage, right, is so ideally to produce correctly assembled bispecific antibodies, right? So the key is that you will have a balanced expression level among the different chains. But this is the only ideal case scenario, right? So in the real practice, so this is very difficult to reach. So that's why we want to try different chain ratios, so that we can get a balanced expression. So the ideal, so in the end we'll have 1-1-1-1 actual expression level of the 4 different chains. And in terms of how we actually perform the chain ratio, right? So we'll do different, like we'll based on 1-1-1-1 ratio, we'll try like a different amount of heavy chain, like a higher amount of heavy chains, and also a higher amount of light chains. So based on the initial purification result in combination with the QC specific, especially the mass and the SEC-HPLC results, right? We'll get an idea of the relative expression level of the different chains, and then we'll choose the ideal one for the scale up.
All right, thank you, Dr. Wang. So for the next question from Cindy, for the CH1CL Crossmap, is the HC1, HC2, and LC2 and LC2 unwanted species clone related or common phenomena across all the CH1CL design? Thank you.
Well, first I will say that it's not related to the design. So ideally, so based on this Crossmap CH1CL crossover technology, right? So ideally, there should be no side product, but as I said, the expression level, the balanced expression level is the key to a successful successfully assemble the Crossmap, right? So in this case, I will see that so we observe an overexpression of LC2 as compared to LC1. So in this case, we have different strategies. One, if the imbalance is not very serious, and we can try to purify based on this product using as shown in the case study, based on their differential binding affinity to the purification resins. And the other strategy is to start over with a different transfection ratio, namely using a higher amount of LC1 than LC2.
All right, thank you, Dr. Wang. We'll move on to the next question. CrossMab technology is from Roche, is under patent. How can you produce proteins using CrossMab? And could you clarify the relationship between Roche and GenScript regarding to CrossMab patent?
Well, so yes, so it's under Roche's patent. So we don't deal with the patent. So it's basically client who would like to use this technology that they have to deal with the patent. So we simply take the sequences provided by our client, and then we will do the production using our TurboCHO expression platform. So we have no relationship with the Roche.
All right, thank you, Dr. Wang. So the next question, what is the timeline for a production of bispecific antibody?
So when you compare the production of bispecific antibody with the typical monoclonal antibody, right? So basically the timeline should be similar, except that if there are some complicated design, then the gene synthesis part will take a slightly longer time than the typical antibody. So, and also if you require multiple step purifications for most of the bispecific antibodies, right? Then it will take 1 or 2 days longer than the typical antibody.
All right, thank you, Dr. Wang. Next question, what optimization steps are taken if the antibody is not expressed in TurboCHO? Does it happen? And the original sequence, is it derived from mouse hybridoma, which is purified monoclonal antibody can be obtained? Gene is codon optimized and synthesized, and standard signal secretion used, but there's no expression.
So it's very rare that you observe absolutely no expression from our TurboCHO expression because we always do a positive control while doing the expression, and in most cases, if there is indeed no expression, we would see that it might be caused by the sequences of the antibody itself. And we will of course do some troubleshooting, and we will look at if client agrees, and we will look at the sequences, and we will pick up some common features that might affect the expression that might cause the expression issue. And in terms of the original sequences, right? So they can be derived from hybridoma, and they can also come from like other antibody discovery technologies like immunization, like from rabbit or fish display or yeast display. They can come from different sources. And some clients even they took commercialized antibody sequences and led us to produce.
All right, thank you, Dr. Wang. Next question. Does bispecific antibody just happen for IgG4 or not?
Well, bispecific antibody, so it's naturally occurring for IgG4, but what we see from our experience that so majority of the bispecific antibodies were came in the form of IgG1. So as I said, it's actually a synthetic molecules rather than a naturally occurring antibodies. And indeed, IgG4, because of the feature of human IgG4, right? People are taking advantage of this feature to generate bispecific antibody platforms, which is called a controlled Fab arm exchange.
Right, thank you, Dr. Wang. The next question, what are the conditions for the creation of for bispecific antibody from upstream process or downstream?
What are the conditions for creation? So first, creation of bispecific antibody. First you need to start from the sequence design, right? So you can take two monoclonal. The sequences can be taken from two monoclonal antibodies, and you reconstruct these two variable regions into one molecules using different formats as shown here. So it can have only one chain, I'm showing here, and it can have multiple chains. And so I would see the creation of the bispecific antibody starts from the design of the sequence, and then followed by the upstream expression and the downstream amplification. And also the QC has to be optimized for different formats as well.
All right, thank you, Dr. Wang. Next question, what is the half-life of bispecific antibodies?
Well, so the half-life, so the Fc region, we know that for a traditional monoclonal antibody, right, the half-life is determined by the Fc region. So it actually also applies to bispecific antibody. If you are looking for a longer or a shorter, or if you are looking for a longer half-life, right? So normally a Fc is suggested to be included in the design of your bispecific antibody molecule. And also there are certain engineerings on the Fc region that can help to extend the half-life. And on the other hand, the lack of Fc region may lead to a shorter half-life. And also there are some strategies that you can actually attach, or you can actually fuse a certain domain to your desired sequences. For example, it can bind to the human serum half-life. So it has no a therapeutic function, but it can bind to the serum albumin so that it can extend the half-life extensively. So there are different strategies.
All right, thank you, Dr. Wang. Next question, does GenScript provide RPLC as part of the standard QC after your turbotrol expressing system?
Okay, I believe that you are referring to the ICLC result demonstrated in the case study, right? So actually, this result was extracted from the LC mass. So we have the RP, the reverse phase column, as the upstream of the mass. So typically for our typical QC analysis, right? SCC HPLC is the default technology. So we do offer RP, but it's not commonly ordered from clients.
Right. Thank you, Dr. Wang. Next question. Can I check for high expression of monoclonal antibody may lead to more bispecific antibody?
Well, if the two monoclonal antibodies, if the two original monoclonal antibodies that you used for the generation of bispecific antibodies, right? If they are if they are high expressor, so they will of course they will certainly benefit for the production of bispecific antibody in terms of the upstream, and this is very beneficial so that you will have a higher yield for the downstream application. And we do have some cases that one of the two monoclonal antibodies is very low expression and the other is relatively okay. And then in the end, in the end, the production of the bispecific antibody has a lot of problems. First, the expression is very low, and second, you will have a very imbalanced expression of these two moieties, which will lead to homodimers and half antibodies, this kind of impurities. So it would generate a lot of difficulties if you have low expressors. But if you start with high expressors, it will give you a lot of it will make your life much easier.
All right, thank you, Dr. Wang. So we'll move on to the next question. Does the 1 IgG SCFV model can influence on the effector functions?
So this is the one showing in the screen, right? Can influence on effector functions. So again, the effector functions relies on the engineering of the Fc region, right? So I wouldn't see there's much influence with or without this Fc region, as long as you keep the Fc region consistent. And again, if you want to modulate the effector functions, there are multiple engineering strategies within the Fc region.
All right, thanks, Dr. Wang. Next question. Regarding the charge Crossmap, how do you control the charge and how many amino acids you need to swap?
How do we control the charge? First, we have knowledge of the orthogonal. We have all knowledge of the amino acids that have charges. So basically, so there are asparagine, lysine, glutamic acid, glutamine, and arginine, and these charged amino acids, right? And from the structural knowledge, we will identify the locations of these amino acids, and we'll see the charge interactions between the different domains, and then you can identify which amino acids you want to use it for the engineering. And of course, there are a lot of applications in this topic as well. And from the glofitamab, as showed in case study 1, so 2 amino acids on each of the domains were utilized for this kind of engineering. And in terms of the swap, right? So the whole domain of the, so you are basically swapping the whole domain. So for example, VH VL crossover, you're basically switching the whole VH with VL. And in terms of CH1CL, you are swapping CH1 and CL completely.
All right, thank you, Dr. Wang. Next question. Why IgG1 is prone for bispecific antibody and which part of IgG1 is responsible for it?
Why IgG1?
Why
Well,
IgG1 is
so,
prone? Yeah.
okay. Oh, yeah. Thank you, Cassie. I got your question. So, I believe that most of the therapeutic targets of this antibody will be used for human, and maybe that's why. So even though on the discovery stage, the origin of the antibody was from mouse or other species, they will be humanized like into human IgG1 sequence. And I hope I've answered the first question. And which part of IgG1 is responsible for bispecific antibody? So for the specificity, right? So it's the variable region of the antibody. So heavy chain has 4 domains. For human, let's take a human IgG1. So human IgG1 heavy chain has 4 domains. Variable domain followed by stage 1 and stage 2 and stage 3. So the variable domain is in charge of the specificity in together with the variable domain of light chain. Yeah. So basically, you are so when you're talking about the specificity, you are looking at the variable region, and the constant region are in charge of the, for example, the effector functions, and so they are important for the serum half-life, and of course the overall stability of the structure of the molecule.
All right, thank you, Dr. Wang. The next question, could CrossMap technology used in other expression CHO system, for example, like XP-CHO?
Yeah, of course. They can, yeah, so there's no restriction in terms of the expression systems. Yeah, we can even express them in like a hexagonal cells. But CHO, so from our experience, our turbo CHO has a much higher expression yield as compared to hexagonal systems.
I see. Okay, thank you, Dr. Wang. I think this is the last question from the floor. Do you have experience with Lonza bispecific antibody platform, which is, I hope I'm pronouncing it correct, BYLOK? If yes, between this and CrossMap, which one is better in terms of manufacturability?
Well, unfortunately, we don't have much experience with the bispecific antibody platform. Yeah, but thank you for mentioning it. I'll take a further look into it.
Okay, thank you, Dr. Wang.
Yeah, but basically, so basically, as long as you provide your analysis, and we will do a thorough evaluation for the sequences and propose the best the optimal strategies for the production. So, yeah, I believe that we have no problem with such different formats.
Okay, thank you, Dr. Wang. I think that's all for the questions from the floor. So for all our audience, if you have any other further questions after this webinar, feel free to email it to us and Dr. Wang Sai. We will answer them via email as well to all of you. Okay, so yeah. So since we have come to the end of the webinar, I would like to express my heartfelt gratitude to everyone for joining us today. It has been truly an insightful and an engaging experience, and I hope you gain valuable knowledge and inspiration from our distinguished speaker for today. So I also wanted to extend our deepest appreciation to Dr. Wang Sai for your outstanding presentation, and we are grateful for your generosity for sharing your knowledge. I'd like to remind everyone that today's webinar has been recorded and the recording will be made available to all of you in the coming days. And this will be an excellent resource for you to revisit the content and share it with your colleagues who couldn't attend this this webinar. So thank you once again for joining this webinar, and hopefully we'll see you again in our next webinar. All right. Thank you, everyone, and goodbye.
Thank you. Bye-bye.
Thank you. Bye-bye.